Okay, now we are going to look at the structures. So this is a prokaryotic cell. It's not this big in size. This is just for educational purposes. In the middle, in this region, it has nucleoid. That's where the DNA material is. There's no nucleus. It looks like nucleus, but there's no actual membrane around it. So there's no actual membrane around it. This is called nucleoid. There's no nucleus, and this material is called nucleoid. So that's one thing that differs. There's no nucleus in prokaryotes. And then there can be plasmids. If you remember what plasmids are, we talked about plasmids when we went over, how you can create transgenic organisms. You can take these plasmids, they're circular DNA that can self-replicate, and you can insert these plasmids into, humans, viruses, using viruses in humans and animals, and you can get the gene of interest, put it in plasmid, and that plasmid can go wherever it needs to go or wherever you want it going. So it has plasmids. They're circles of DNA that can self-replicate. And there is something called pilus. Pilus is a hair-like structure like this. It can help it hold on to stuff. So hold on to surfaces, adhere to objects. And there is also something called sex pilus or sex pili. So using this sex pili, it can exchange genetic material. There is no male or female, but it can exchange genetic material and organisms. I don't know if you guys heard about MRSA. So organisms that are antibiotic resistant, they have a resistant gene, and if this exchange those genes with another organism that doesn't have that gene, then it's going to make that gene also antibiotic resistant. So this is how they become antibiotic resistant. Mainly they can exchange genes using sex pili, and then it'd be very hard to kill it. We might have to develop another antibiotics if it becomes resistant to it. And that's how those MRSA organisms come around. They're resistant to antibiotics, and then you have to develop a new one, which can be very expensive. Then there is cell wall. Cell wall is going to give it shape. Another thing cell wall does, it provides protection. CDC says that you have to boil your water for 20 minutes or whatever food you're eating. At least 20 minutes of boiling is going to kill that bacteria. If you don't boil it for 20 minutes, maybe you do it 10 minutes, this cell wall dries up, but this DNA or genetic material is still there. So if you stop boiling it, it's probably gonna be able to still live. So this cell wall is going to give it protection. Another thing is maybe you are drinking a cup of coffee and then you decide that you want to go on a vacation. You leave that cup of coffee on your counter. When you come back, all that maybe bacteria is dried up, and you think, oh, this bacteria cannot live without water for that long. So that bacteria, what they can do, let me show you, they can make endospores. What endospore is, it can take all that genetic material and it can put like a capsule around it, and it can basically save it until the conditions are right. Means there's water, there's food, there's nutrition, then it can start growing again. So endospores are formed to survive in harsh conditions. Actually says that here. So it helps the bacteria survive in harsh conditions. One bacteria that's around, that you might have heard about is called Clostridium botulism. So botulism, botulinum is the cause of botulism. That's one example. It can form these endospores that can survive really hot temperatures. Even if you decide to wipe the surface down with Clorox or the antibiotics, endospores can still survive. So you might have to autoclave something. If you don't know what that means, it means putting something at really, really high temperature. That might be able to kill endospores, but Something like a Clorox wipe wouldn't be able to kill endospore. Even boiling it wouldn't be able to kill endospore. So have you guys heard about botulism? No? Okay. So if you have a food can and it starts to look like this, that means, so Clostridium botulinum is a bacteria. It's a soil bacteria, and it can contaminate food. So if it starts to look like this, that means it's been contaminated. It's contaminated, and that bacteria is anaerobic, means it doesn't need oxygen. And this can doesn't have oxygen because it's all closed up. And once that bacteria starts to live inside, it makes gas, and the gas makes the can look like this. If you have canned food that looks like this, do not eat it. So it can cause botulism. So that's one example. Also, um, so in honey, there is some soil bacteria that can contaminate it. So if you have, if you have in a baby, do not give the baby honey. Maybe wait until they are a toddler, because honey can have some of this soil bacteria in it that can cause botulism. So it's not safe to give it to, babies, because they don't have their immune system build up, and just wait until maybe they're toddlers. So that's anaerobic bacteria that can form endospores, and it can live a really, really, really long time. Some of fungus can do that too. It can form endospores, and it starts to germinate when maybe it rains down or there is food, and the temperature is right, then it can start to grow again. I skipped the slide, this one. I need you to know 3 different shapes of prokaryotes. There is more than 3, but I only need you guys to know 3. If you take microbiology, you can learn all the other types. One is coccus, cocci. It's a spherical circles. So it's a circular bacteria, or archaea. So if it's in chains, that's where the name strep comes from. Streptococcus is circles that are in chains. If it's a rod shape, it looks like a rod, it's bacillus. And if it looks like corkscrew top or a spiral shape, it's called spirillum. I might show you a picture and ask you what bacteria is this. So I might say you have rod-shaped bacteria, or you look under the microscope, the bacteria looks rod-shaped, which type is it? And I'll give you coccus, bacillus, spirillum, you have to pick bacillus. So just make sure you know these 3 shapes of bacteria. Any questions so far? No? Okay. So prokaryotes, they are very, very diverse. The reason they're so diverse is because they can live on lots of different things. There are autotrophs. Auto means self. Troph, feeding. It's a self-feeding organism, makes its own food. Phototrophs, the one that take sunlight and make food, are type of autotrophs. So it gets all the carbon, whatever it needs, by consuming from sunlight, and that's type of autotroph. We are heterotrophs. We eat other things. We depend on other things. So heterotroph is an organism that gets its required material by eating other organisms. Then there are chemotrophs. They get energy from chemicals. And chemotrophs, they can just live on minerals. So in caves, maybe where there's no sunlight, only rocks are there, they can just feed on those rocks. So they're very diverse. And then, so they can live on those rocks, on the sulfur or the minerals. And then there is obligate aerobes and anaerobes. Obligate means it requires. Obligate aerobes, they require oxygen. If there's no oxygen around, they're going to die. So they require oxygen. Anaerobes are the ones that do not need oxygen. They can make ATP without oxygen. Bacteria in your teeth or in your gut, they're anaerobes. Clostridium botulinum is also an anaerobic bacteria. So they do not need oxygen, but if oxygen is around, that's not harmful either, but they're just not gonna use it. There are something that's called obligate anaerobe. So they do not use oxygen, and if oxygen is around, that's gonna kill them if they're an obligate anaerobe. So these are different ways prokaryotes can obtain their carbon and energy. That's why I think I showed you hydrothermal vent. There is bacteria there that's feeding on these rocks, and it's able to turn some minerals into food for the maybe plants that are growing there. Okay, so in prokaryotes, we've been talking about bacteria. There is something that's called proteobacteria. It's one phylum of bacteria. They're very diverse. There's lots of them. This is single cell proteobacteria. There's multicellular proteobacteria, so very diverse. This bacteria looks spherical, so coccus, and it's in chains. This bacteria is called cyanobacteria. That's something you find in your pond that looks kind of blue-green. Looks like algae, but the algae is made up of bacteria. Two types I want you to know is E. coli in humans, Salmonella in reptiles and birds. So Salmonella is a type of E. coli, but it's in reptiles and birds. So there are two types of proteobacteria. E. coli is found in your gut, so in your poop as well. So it's very important that you don't get E. coli in your food, because then it's gonna get you sick. So E. coli, and also I wanted to mention, I usually talk about this in anatomy. Females, there's a higher number of UTIs that happen in females, so it's more common in females. UTI are urinary tract infections, and that mainly comes from a wiping issue. So there's E. coli in your poop. If you're wiping wrong, it's gonna get into a spot where it's not supposed to be, and then it's gonna cause infection. So as long as there's bacteria where it's supposed to be, but once it gets in a spot where it's not supposed to be, then it can cause infection. So if you keep maybe using hand sanitizer and that keeps killing the bacteria, normal bacteria or beneficial bacteria that's on your hands, it's going to open up the space for the bacteria that's not supposed to be there, and it can cause infections if you overuse hand sanitizer. Salmonella is a type of E. coli found in birds and reptiles. So maybe chicken eggs. So chicken egg comes out of the same spot where the poop comes out. So it's very important to maybe wash the egg. And if you're trying to eat raw egg, maybe wash it, and maybe with antibiotics. And then when you break the egg, make sure there's no egg shells in your egg, or just boil it really, really good. So salmonella is type of E. coli that you can get from birds and reptiles. Those are two types of bacteria that are proteobacteria I want you to know. So going back here, so far we said we have bacteria, and that's a domain. And then for the phylum, we have proteobacteria. And in proteobacteria, we have E. coli in humans and Salmonella in reptiles and birds. This is phylum. This is domain. So making a chart might be helpful if you're trying to keep track of the type of bacteria or archaea species we have covered. So there is cyanobacteria. That's what it looks like. It's bacteria in chains, more than one, so multicellular. That's from another phylum of bacteria. It's not proteobacteria. These autotrophs were the first one to release oxygen. So in early Earth, when these, so the name comes from color. Cyano is a color, blue-green color. So when these cyanobacteria came around first, they were the first ones to release oxygen. There was no oxygen on early Earth. Once oxygen came around, all the organisms that didn't use oxygen, maybe oxygen was harmful to them, they started dying. So that's how the first mass extinction happened, with cyanobacteria releasing oxygen in the environment. They you do photosynthesis, and that's their autotrophs or phototrophs, whichever you want to apply to them. And then that's how they make their food, and they release oxygen in the environment. So that's another phylum. So if we're talking about bacteria here, another phylum is cyanobacteria.